let's continue with the laser. When these excited atoms in the active medium get excited and move to a higher energy level, when these excited atoms return to their lower energy state, they release energy in the form of photons. The photons emitted by the excited atoms stimulate other excited atoms to release photons of the same wavelength and phase, thus generating a coherent beam of light. What is the application of laser? Confocal microscopy. Laser light is used to illuminate the sample in a confocal microscope, allowing for precise high-resolution imaging. Next is the flow cytometry. Laser beams are used to analyze and sort cells based on their physical and chemical properties, in the case of flow cytometry. Then DNA sequencing. Lasers are used to generate the fluorescent signals needed for DNA sequencing. Next is the photodynamic therapy. Lasers are used to activate photosensitive drugs that selectively destroy cancerous cells. Laser surgery. Lasers are used for precise and minimally invasive surgical procedures. What are the advantages of lasers in biology? Before moving on to that, let's have a quick recap about the applications. Used in the confocal microscopy, flow cytometry, DNA sequencing, and photodynamic therapy and laser surgery. First advantage is the precision. Lasers can be focused into a very small spot, allowing for highly precise and localized interactions with biological samples. Sensitivity. Lasers can be used to detect and analyze very low concentrations of biological molecules. Next is the speed. Lasers can generate light at very high frequencies, making them useful for high-speed imaging and analyzing. Non-invasiveness. In many cases, lasers can be used in a non-invasive manner, reducing damage to biological samples. Disadvantages of lasers in biology. Cost. Lasers can be expensive to purchase and maintain. Safety concerns. Lasers can be hazardous to human skin and eye, and special precautions must be taken when working with them. Limited range. Lasers are highly directional, which can limit their utility in some applications. Specificity also there. Lasers emit light of specific wavelength, which may limit their ability to interact with certain biological molecules or structures. Phenomenon of laser. The term laser stands for light amplification by stimulated emission of radiation. The laser is a device that produces a beam of coherent light that is monochromatic, of single wavelength, directional, and with high intensity. The phenomenon of laser is based on the stimulated emission of photons, which occurs when a photon stimulates an atom or molecule to emit another photon of the same frequency, phase, and direction. The basic principle of laser operates include the excitation of a gain medium like solid, liquid, gas, or semiconductor by means of an external energy source such as an electrical discharge or optical pump. This excitation leads to the population inversion of the medium, where more atoms or molecules are in the excited state than the ground state. Then the photon passes through the excited medium. It stimulates the emission of another photon, which is coherent and has the same direction, frequency, and phase as the original photon. The stimulated emission process amplifies the photon flux, leading to the creation of a laser beam. The laser phenomenon is characterized by several properties that make it unique and useful in many applications. One of the most important properties of laser light is its coherence, which means that the electromagnetic waves are in phase with each other and have a fixed relationship in space and time. The laser phenomenon is characterized by several properties that make it unique and useful in many applications. One of the most important property of laser might be its coherence, which means that the electromagnetic waves are in phase with each other and have a fixed relationship in space and time. This property allows the laser beam to be focused into a small spot with high intensity, enabling precise cutting, drilling, and welding of materials. Another property of laser light is its monochromaticity, which means that the light consists of a single wavelength. This property is important in spectroscopy and microscopy, where the specific wavelength of the laser can be used to excite specific molecules or cells, leading to the emission of fluorescence or other types of signals. The laser phenomenon has many applications in biology, including microscopy, flow cytometry, DNA sequencing, cell sorting, and gene manipulation. For example, laser confocal microscopy enables the imaging of cellular structures with high resolution, while flow cytometry allows the sorting and analysis of cells based on their physical and biochemical properties. Laser tweezers can be used to manipulate cells and organelles for studying their functions, while femtosecond lasers can be used to cutting and drilling the tissues without causing thermal damage. In summary, the laser phenomenon is based on the stimulated emission of photons, leading to the creation of a coherent, monochromatic, and high intensity beam of light. The properties of laser light make it useful in many biological applications, including microscopy, flow cytometry, DNA sequencing, and cell sorting. Adsorption, partition, and ion exchange chromatography, principal working and application advantages. Now we are moving on to the chromatography portion. Chromatography is a widely used technique for separating and purifying different molecules in a sample. There are various types of chromatography techniques available, including adsorption chromatography, partition chromatography, and ion exchange chromatography. First, we are going to learn about the adsorption chromatography. Adsorption chromatography is based on the adsorption of molecules onto a stationary phase, such as a solid matrix, and their subsequent separation based on their interaction with the stationary phase. The mobile phase, which contains the sample mixture, is allowed to flow over the stationary phase, and the different components of the sample are separated based on their affinity towards the stationary phase. In adsorption chromatography, the stationary phase may consist of a solid matrix such as silica gel or alumina with varying pore sizes and surface charges. The sample is applied onto the stationary phase, and the mobile phase is allowed to flow over the stationary phase. The components of the sample interact with the stationary phase and are separated based on their affinity towards it. The more strongly a component interacts with the stationary phase, the slower it will move through the column. The separated components can then be collected and further analyzed. Adsorption chromatography is widely used in biochemistry for the purification of proteins, nucleic acids, and other biomolecules. It is also used in pharmaceuticals and chemical industries for the purification of various compounds. Next is the partition chromatography. Partition chromatography is based on the differential partitioning of molecules between two immiscible phases, such as liquid and gas, or two different liquids. The stationary phase consists of liquid or solid support coated with the liquid phase that is immiscible with the mobile phase.